Hello, we are going to first review DNA replication today, and then we'll move into notes. So the first question is, what's the name of the enzyme that unzips the DNA? This would be helicase. So helicase is the name of that enzyme that unzips the DNA. What's the name of the enzyme that adds the new nucleotides to the DNA strand? That would be DNA polymerase. What can we say about the new DNA molecules that are made? You see pictures there. Maybe you wrote that they are identical to each other or identical to the original, or they have half of the original strand and half of the new strand. All of those would be correct. Where do the nucleotides for the new DNA come from? So they come from the food we eat or from old cells that have been recycled. All right, we're going to skip this one, but in order of the images, you can take a second and think about what order would these images go in. So we're going to start right with this one right here. Helicase unzips, so this one's going to be next. We're adding the new nucleotides. This will be 3, 4, and then 5. All right, so in your notebook, your title for today is going to be proteins. We've been talking a lot about how DNA has the instructions on how to make proteins. Now we're going to talk about what the heck a protein is. Here's a computer image. They make it nice and beautiful and rainbow colored. They're not really like that in real life, but this is just one representation of a protein. So our goal for today is to describe the structure of proteins. So what are the building blocks? What are the bonds? What's the shape? All of those types of things. Take a look at these amino acids. Tell me, how are they similar? How are these pictures of molecules similar? So you'll notice this top part is similar. So maybe you said, oh, they have N's or they all have C's, or maybe you got a little bit more specific. This top part is all similar between these things called amino acids. And you might be asking, well, Ms. Frankson, what the heck's an amino acid? An amino acid is a building block of a protein. So let's write that down. Amino acids, they are building blocks of proteins. There are 20 different amino acids. We saw some examples on the previous slide. But think about like DNA. DNA also has building blocks. The building blocks are nucleotides. There are 4 different building blocks, right? There's a building block that has an A for a base, a T for a base, a C, and a G. With amino acids, we have 20 different ones. There's a similar structure that all these amino acids have. We saw this on the previous page. See, they all have this top box is the same. So I'm going to have you draw this in your notes. Go ahead, pause the video while you draw this in your notes. And we're going to label this. Okay, so why the heck is it called an amino acid? Because the N and the 2 H's, this is called the amine group. Scientists like to name groups of letters in molecules. So if it has an N and 2 H's, that's an amine group. And then there's an acid group. Okay, the acid group is the C with the O and the OH. So go ahead and label these on your drawing. If you would like to circle the N and the 2 H's and circle the C, O, and the OH, that is your acid group. You'll also notice there's this R down here. We'll talk about that in a second. So if you need more time, go ahead and pause. Otherwise, I'm moving on. So the R group, this part down at the bottom, scientists are lazy and they write R for rest of molecule because this is what's different on each amino acid. So what I need you to write down in your notes is the R group is what makes each amino acid unique. And so each amino acid has a different part here. And scientists just write an R to show that that's the rest of the molecule. So amino acids, building blocks of proteins. We stick a bunch of amino acids together, we get a protein. Okay, so here's some examples of those R groups, right? And so notice the bottom part is different on these different amino acids. So how do we connect them together then to make a protein? Go ahead and write this down. Peptide bonds are the bonds that connect amino acids together. We talked about hydrogen bonds in DNA. There's a different type of bond here called a peptide bond. You'll see in this picture, we've got this amino acid on the left and this amino acid on the right, and they're joining together. So take a look at all of these. This is just another way to draw an amino acid chain or a protein. So these are all amino acids, and in between each one of these, there would be a bond, a peptide bond. So if I had 5 amino acids here, how many bonds would connect them? If I wanted to connect them all in a line, we would have 4. 1, 2, 3, 4. So you're always going to have one less bond than the number of amino acids that you have. So what the heck do protein molecules actually look like? You don't actually have to write anything down here, but here's a couple different models. Remember, they're made of C's, H's, and O's, but eventually those make shapes, okay? And so these are different computer models of proteins. Proteins kind of look kind of crazy and complicated. You do need to write this down. Proteins can be lots of different shapes. DNA can't be different shapes. DNA is a double helix all the time. But proteins can be lots of different shapes because they do lots of different things in our bodies or in an organism. The shape determines what job they do. Some of them are long and skinny. Some of them are short. Some of them are made of 2 parts. We're going to talk about some examples of proteins in the next couple of days, but all of these proteins are different shapes because they have different jobs. And so if we have a problem with the protein or a problem with the code of DNA that's coding for that protein, maybe our protein's not going to be the exact right shape and it's not going to be able to do its job. So an example of a protein, we've talked about this before, but the spike protein on SARS-CoV-2, the virus that caused COVID-19, this is how it gets into our bodies, okay? So it's got different spikes on here, and these spikes are how it attaches to a cell. So these are the spikes again, and the spike binds to a receptor, and then the receptor says, okay, you can come into the cell, and then it allows the virus in, and then the virus can make us sick. So that's one example of protein. We've also talked about how proteins make channels in our cell membrane. It's another job of a protein. And we're going to talk about some more examples tomorrow and the next day.